Some definitions. For the people in the audience, we don't use microflora anymore, okay? We know they're not flowers, they're a group of their own, they're the microbiota. If you see the term microbiota, that means who's there and their relative abundance, as Jas said. Microbiome, that's the functions they bring to the party, okay? What they actually do in the human body. And then we have these omic technologies that we're developing and applying to this system. And that's metagenomics, which is grabbing a gene catalogue from a sample and telling you what functions are there. We have metataxonomics, which is a neologism I coined because there is confusion between metagenomics in the literature, especially in the clinical literature, and that's where we're using this surrogate gene to determine the presence or absence of an organism in a sample. And that allows us to do very high throughput analysis of many samples. And then we have metabolomics, or sometimes called metabonomics. And this is the ability to capture a snapshot of the metabolic profile of a sample. So try to keep those terms in mind. So the main focus of the human biology in the last sort of century was this sort of paradigm here. You inherited your genes from your parents, those interact with the environment, and that leads to disease and health. And what we've realized in the last 10 years, and I'll stress this, as microbiome people, we've only been at this really full on for the last 10 years, is that the microbiome is an environmental factor that interacts with your genome, interacts with the environment, and translates to a health disease outcome. And unlike your genome, we can modulate this, we can change it. It's also host specific, so everybody in this room has their own microbiome and their own microbiota, which reflects the natural history of you as an individual. Whether you were cesarean or natural birth, whether you were breastfed, whether you're a twin, these are all factors that are found in your microbiota. You have a unique microbiota, and in this room we may only have 1 or 2 species that's shared in all our guts. Okay? Now, lots of the literature at the moment is based on what Jas said is stool samples. Also think that the niche in the large intestine is totally different to the niche in the small intestine. The small intestinal microbiota is not the same as the large intestinal microbiota. So trying to say what we see in the stool sample is happening through the whole digestive system, step back and think it's different to the small intestine, okay? So a lot of the literature you see is based on stool samples. For obvious reasons, they're easy to get, well, relatively easy to get. We can change the microbiota by diet, by antibiotics, and by surgery. Whether you actually want to use and do this is another question. I'll come on to that in a moment. So one of the key findings that I want you to take home from this is studies into sterile animals, notobiotic animals, or sometimes called germ-free. If we remove the microbiome, every single aspect of that animal is impacted by the absence of a microbiome. So that leads to a fundamental understanding that mammals have co-evolved a requirement for their microbiome, and not the other way around. Bacteria don't need mammals. Mammals definitely need the microbiome in order to develop properly and to function properly. And for this audience, that means that they have smaller livers and increased ferritin. I don't know the significance of that. I'm not a clinician. I'm a microbiologist who's interested in understanding how the microbiota supports ecosystem function. I'm an ecologist as well. But this is a take-home message. The absence of a microbiome impacts the whole host. It has a global impact, in other words. Now, evidence in the literature in the last 10 years has been a lot of excitement to grab a plot of land in this gold rush and establish some link between what's in the large intestine and some of these big diseases. Now, as it's already been mentioned, correlation is not causality. At the moment, we're undertaking lots of observational studies. And what I'd say to you is, as clinicians, if somebody came up to you and said, we've just found a new immune system or organ in the human body, where would you expect us to be in 10 years? We would still be looking at this system and trying to determine what's correlated, what's variable. And that's what we're doing at the moment. We're trying to understand the variability in this system by observing it. So what we're trying to do is get away from the correlations and ultimately find the causal links between what's happening in the large intestine and these diseases. So the microbiota in the large intestine and in the small intestine and the other microbiotas around the body do impact at a global level. Also, they impact at a local level. So we've shown that colorectal cancer, the microbiome on a tumour is different to the microbiome just off a tumour. And we think that there's a hypothesis here that off-tumour microbes are driving the first hit on the oncogenes that lead to tumour development. Then as the tumour develops, the microbiota changes on that new environment. So they can have a local impact as well. And moreover, which is relevant to you guys, they can also have an impact on the drugs that you give your patients. They will modify the response of the individual to those drugs. So these microbiota integrated into clinical thinking is the way forward as we see it. So, the dialogue in a healthy individual, that's what we're interested in at the moment, establishing this. This is between the metabolites and the proteins that these microbes make. So that's what we really want to understand, the metabolites, protein, the dialogue, and how that's influencing the host. So what do we want to know? Okay, we've only recently discovered the microbiome is important to human biology. Still in observational phase, we want some fundamental questions. Are there a common core set of genes that everybody has, functions? So far we can say yes for some examples. One of the organisms that Jas was talking about, they produce short-chain fatty acids. We have evolved receptors to short-chain fatty acids. Short-chain fatty acids are only made by bacteria and it doesn't come in your diet. So there is a requirement for us to understand how the short-chain fatty acids interact with the host. Are there common species in the microbiome? There are if we take everybody's guts. Each individual has about 160 species in your gut and several thousand strains. But everybody in this room should have most probably levels of Faecalibacterium prosnitzii. So that's a common organism. How can we modulate the composition? If we want to treat it as an organ, how do we change it? What happens if we modulate or change it? And do we actually want to? So giving people rifaximin, is that a good idea? Given an antibiotic that causes collateral damage in your gut microbiota, is that good long-term down the line? So a lot of people working in this area came from the environmental microbiology setting. And environmental microbiologists and microbiologists in general do not put our hands up and say we're very poor at growing organisms in the laboratory. In the human body, we're pretty good at doing it. If you're unfortunate to work in the soil system, 99% of the organisms in a soil system cannot be grown in the laboratory. So what we did is we developed DNA-based, nucleic acid-based, or culture-independent methods in order to understand who's there, their abundance, and what they're doing. And we brought this baggage and prejudice into the human microbiome area, and so that's why we have all these different omic approaches in order to understand who's there. Metagenomics grabs all the genes in a sample, produces a gene catalogue. This tells you who's there, relative abundance. This tells you what metabolites are there. Those in green are also methods, but they're not so well developed and technology is not so robust because they require good bioinformatic information. So we have a bottom-up and a top-down approach, and that's based on the genetic flow of information. What we're really trying to do is bring together methods. So we've been developing metataxonomic methods that tell us who's in their abundance and linking that directly in correlations to metabolites. We're also bringing in something called degradomics, and I didn't think of this word, but it's looking at proteases in the gut. So, marrying together omic technologies, what do? We create matrices of patient samples and their metabolites, and then same patients from the same samples and the species of organisms. We cross-correlate and we produce like correlation matrices, which we can then show as a heat map. And here we have an example of bacteria 2 correlating very strongly with metabolite 1. Or this could be a function, it could be an enzymatic function, like bile processing genes. So this is the way we go forward. We take our samples, we create these large omic data sets and then correlate them. Once we have strong correlations, we move forward into causal links. So I want to talk about how we've used this as an example, a vignette here, in bariatric surgery. So bariatric surgery, this is the Roux-en-Y gastric bypass, which is the gold standard for treating individuals with BMIs of 30 or more. Works really well. We had a rat model of this and we want to understand, because in these individuals with type 2 diabetes, when you give them this surgical intervention, within a week a significant proportion get rid of their type 2 diabetes, and that's not due to calorie restrictions, it's due to some other mechanism. So we wanted to ask the question, could it be due to modification of the bacteria? Could it be due to modification of metabolites? So we took our rats, we gave them RYGB, not because they were fat, they didn't have a choice, but we looked at metabolite profile. So this is what principal component analysis Jess was talking about. Each spot here represents 1,000 variables. Because the human brain can't cope with 1,000 variables, we reduce it down into two-dimensional space. So these are all the rats that have had the surgery. These are all the rats that were sham, opened up, their guts were pulled out, put back in, but they didn't have the bypass. And you can see that the metabolite profiles of the two separate. In urine, you're looking at the metabolites within the host. In feces, you're looking at the metabolites due to the bacteria mainly. And then we look at the bacterial composition, same sort of principle. One dot here may represent 100 different species. Any two dots close to each other shows the same type of pattern. The microbiota is altered. When we mine down into this in more detail, what we see, and looking at these pie charts down here, is a strong enrichment of gamma proteobacteria that ultimately translate in huge amounts of E. coli in the intestines of these rats. This is the same as in humans, and this is stable for about a year. These individuals have high levels, sometimes up to 40 to 50% of their fecal material is E. coli. And E. coli, my hypothesis, is not an organism that lives in the small and large intestine. It's a small intestinal organism that just happens to spill over into the large intestine and just about cope living in there. If somebody came in to me with a fecal sample that had that level of E. coli in them and they hadn't had RYGB, I'll be telling them to go to see their GP because they've got something seriously wrong with their gut. This modulation of the gut microbiota by the surgery also changes a whole suite of metabolites. And these are just some of them that changed. We were really interested in the trimethylamines, okay? These are microbial metabolites made from choline and ultimately made from phosphatidylcholine in the diet. This was interesting because a PNAS paper had linked these to cardiovascular disease. But what I want to show here is it's not as simple as this, and it's never as simple as this. This is a multifactorial system. When you look at their epidemiological data, these individuals' cardiovascular risks reduce, so they get better in that respect. So it can't be as simple as TMAO and TMA linking to heart disease. Okay, so we started looking then at the whole environment. If you've got lots of E. coli in there, you're producing lots of endotoxin. And just to mention here, not all endotoxin is equal. Bacteroides make lots of endotoxin. We don't know the impact of that endotoxin compared to E. coli's endotoxin compared to salmonellas. But we do know in our RYGB animals is that this environment is so cytotoxic we couldn't actually measure it using the OECD lymphoblastoid measurement. It's too cytotoxic, but it's not genotoxic, so it doesn't damage DNA, but it does cause a lot of death in the cells. And interestingly, these individuals do have a higher risk of developing colorectal cancer. So in this example here, modulation of the gut microbiota may be causing short-term gain, but long-term pain and inflammation in the gut. Okay, so we also then looked at bile acids as a mechanism maybe for altering the type 2 diabetes. And we can see that in the rats there is a dramatic change in the bile acid profiles. But we don't know whether or not that's driven by the microbiota or is driven by the surgery. But one thing that's interesting is that a paper out by Watanabe have shown that if you combine bile acids in the gut and in the host, it improves insulin resistance in metabolic syndrome. So the bile acids are playing a role here, and it goes back to FXR, in type 2 diabetes potentially. Whether or not that's modulated the gut microbiota, we now need to see. And we're taking organisms out, incubating them with the bile acid to see if we can recapitulate the patterns that we're seeing in these sets of data. So that's quickly looking through that in terms of using multimodal omics. Now what I want to bring finally to you is the idea of actually measuring good old-fashioned functions, enzymatic function in a fecal sample. So while we're interested in who's there, that's always one of the questions we're asking, we really are interested in what they're doing. And it seems that the gut or the human body doesn't care who brings the beer to the party. In other words, as long as that beer is coming to that party, it doesn't matter who brings it. So as long as there's short-chain fatty acid production in the gut, doesn't matter which organisms are producing it. It just needs to be there. Proteases are a similar sort of story. It seems that we all have fecal proteases or bacterial proteases in our gut. So we're interested in this because bacterial proteases are potential virulence factors in colorectal cancer and inflammatory bowel disease. And we were looking at inflammatory bowel disease in this model. And nicely, Steck et al., which is Dirk Haller and Balfour Salter's paper, had shown that this protease here from Enterococcus faecalis compromises tight junctions. As was shown before by Wim, if you take a transwell, you put this jelly in here, the cells come apart and fluorescein can flow from the upper compartment down to the bottom compartment. So jelly, the protease, compromises tight junction integrity. This was their model. So this is a pathogenic organism. It's producing a protease called jelly. It messes about with tight junctions. It allows everything to move across. We were interested in asking the question, in inflammatory bowel disease, whilst proteases are interesting, a lot of people are focusing on host matrix metalloproteases. We wanted to ask the question, could it be the microbiome and its proteases that are driving this? So we started looking at a group of healthy individuals in green here and a group of IBD sufferers in red here. And we were measuring the integrity of tight junctions in a colonocyte model in transwells using fecal water from these individuals that contain fecal proteases that were bacterial in origin, and we normalized for total proteases in the samples. And what we're looking at here, here's our jelly here, and as you go lower down here, that means the junctions are coming apart more. The transepithelial resistance is dropping. So this is our control here. So you can see some individuals, there's quite a lot of variation. But interestingly, I think this is the more interesting thing, is that the healthy population showed quite a large amount of variation as well. And if we give them bacterial protease inhibitors, we can more or less normalize and bring that down. This is what's interesting me here, not so much this at the moment. The fact that in the healthy population we have quite a range of fecal protease levels, that when, I'll ignore that moment, that when we actually plot against tight junction, we can get a nice relationship that shows that high fecal proteases, which are bacterial in origin, actually leads to leaky junctions. So we're interested in taking this forward and thinking further that in a healthy population, I can take this room here and I can divide you into a continuum of different types of organisms in your gut, different types of endotoxins, and different levels of fecal proteases that are coming from your bacterial or your microbiome. And incidentally, low exposure to LPS does lead to inflammation of the liver. So everybody sits on this continuum. It's not as clear. Some individuals have lots of bacteroides and some individuals have lots of firmicutes. That'll lead to different levels of proteases. We know those proteases can interact with your tight junctions to cause them to become leaky. At the same time, different individuals will be producing different types of LPS, either bacteroides driven or maybe E. coli driven. This can then lead to leakiness and exposure of your liver to endotoxin, potentially leading to NASH or not. So depending on where you are on here, you might have propensity to have this or propensity to have this. Of course, it's more complicated, the fact that intestinal alkaline phosphatases can also detoxify that. It gets even more complex that these high fecal proteases can actually cause inflammation of the gut itself. Furthermore, and to rush through this, the bacteria make alkaline phosphatases that can also detoxify LPS, lipoteichoic acids, and flagellin, various endotoxins. And that leads to a model then, depending on where you are in this continuum, you could be in a situation where you have inflammation of both these organs here due to being unlucky because you inherited the wrong types of bacteria. And we can try to then modulate this to push you over to this state here. So, conclusions. We're really at a new chapter in human biology. And what you need to take home away from this is the fact that you need to think about the microbiome as an environmental factor as you would with diet, drinking, whatever it is, in understanding how your patients function. It plays a fundamental role in mammalian biology. We don't fully understand the mechanisms, and that's where we're going at the moment. We have a very good omic toolbox that can allow us to interrogate this system, but we need to explore the ramifications of what this is really showing us. And we really need to start to link the microbes to the metabolites. Okay, we need to be able to say that this microbe makes that metabolite, and that metabolite is bioactive and affects this pathway, either inflammatory, anti or pro. And with that, I thank you for listening. I thank all my colleagues and funders for funding me and allowing me to be here to present this work. Thank you.